Thank you very much. So also welcome from my side. So I'm Hans Sanden. I will do this presentation together with Akos and Paval here at Boko. We also work with the University of Porto and S4 mobile laboratories. So we are looking at in situ subsurface scatology. No, next slide, Akos. So there is a lot of carbon in the soil, as you know, and especially actually here in the temperature and boreal regions, there's a lot of carbon. Um, Great, but our interest is it getting less and getting more? So next slide. Is there room for more carbon in the soil? And where we really can affect the amount of carbon in the soil is the soils we're actually working on a yearly basis. And this is actually only the agriculture soils. Forests we touch now and then and so on, but agriculture soils we're working on every year. So if we can increase the carbon in the agriculture soil by changing our methods, this can actually have a big effect. And there are studies showing that, especially with minimizing tilling and so on, we can actually increase up to 200 kilos of carbon per hectare in year. And the EU, they have this thing about 4 per 1,000 project. So increasing the amount of carbon in agriculture soils by 4 per mil per year would actually solve a big part of the problem. And we're on the soils every year, so this is where we have a chance to do it. But next slide. How do we measure carbon stocks? Well, so far, when doing all the models and so on, we've actually been using space for time. We've been looking at this forest was turned into cropland 15 years ago. How much has the carbon changed? What's the difference between the forest and the cropland next to each other, essentially? And if we plant trees on it again, what happens? And yeah, this has been doing in Ethiopia, where there's very little data, of course, on carbon stocks. So it brings a very good knowledge about potentially what's going on. And next slide. And then we can use this for doing modeling. So in this case, it's not agriculture, it's actually forestry. But we can see using different management methods of different thinnings will actually affect the incorporation of carbon into soil greatly. And it's the same thing in agricultural soils. But for the farmer, this is way too coarse. For the farmer to know if he's accumulating soil or not, we need to do it on a much smaller scale. We need repeated sampling. So far, what we've been doing here, for example, together with Argus, is that we've been repeatedly going back to the same plot, taking soil samples, analyzing it in the lab. And what you see is that with minimum tilling, for example, you get an increase in the top layer. But if we look at the deeper layer, next slide, we can see that actually is no effect. And what's happening below that, we don't really know, because it's so hard work to actually sample in deep soil depth and doing all these analysis. So what we need is a quick way and a cheap way to analyze carbon stocks changes. So, Akos.
Yeah, so thank you, Hans, for your introduction about carbon stocks and the importance of them. Now, for the start, we will look at currently available technologies to monitor soil physicochemical properties, including organic carbon, and give detailed overview about one of them. In this paper, published just recently by Schmidinger et al., the performance of different type of soil sensor was compared. Not surprisingly, near-infrared and gamma-ray spectroscopy demonstrated to be the most effective, both as single sensors or in combination with other sensors. So from further on, we will explore the capabilities of visible near-infrared spectroscopy used in soil sciences. To build it down to basic steps, we apply in our methods, we start with a selected soil sample. In a standard way, when doing it ex situ, this can be a 90 centimeter long soil core. From this, we record spectra, looking at every single centimeter going down from the top to the bottom. After the acquisition of spectra, we process our soil samples in the lab. So we achieve target soil variables, also called ground truth data. For example, water content or organic carbon. Now, with the combination of the spectra and the ground truth data, we have a full database ready for modeling. So we can apply a linear regression model for a given element. Nowadays, an increasing amount of research is focusing on applying these kind of methods on large soil databases observed in situ. One of PHENET's main goals is to bring in innovative automatable techniques, such as the subterranean green from S4 mobile laboratories. This is a mobile field unit carrying a visible and a near-infrared spectrometer and a load cell for measuring probe insertion force. It works like it goes down to the soil, centimeter by centimeter, measuring the light reflected from a column of a soil adjacent to the probe. As Hans mentioned before, this was our first real testing site, so this tilling experiment in Fuchsenbiegel here in Austria. You can see there was a randomized block design of 3 different tilling methods, so conventional, reduced, and minimum tillage. From every single of these methods, from this plot, we collected like 15 points. So first, the tractor went and collected the soil cores. These soil cores were selected in different layers and kept in separate bags to later do an analysis in the lab. After the tractor, Paval came here, my colleague Paval here with the Subterra, and he recorded the spectra exactly at the insertion point of the soil cores, or like maximum 20 centimeter away. And we also collected our soil and take it back to the lab, and we captured ex situ spectra on it. This is also very important. I will say it later. And with the combination with the in situ and ex situ data, we were ready to start modeling. I also wanted to mention, like, the creating of a digital twin of our experimental area, which I just showed you. So here you can see the interpretation of this in Quantum GIS, how it looks like. Yes, now we will look more into some spectra. There are a lot of challenges when we are processing our data. One of them, arguably the most important one, is the water effect. Spectra with varying soil water contents, even if they are the same soil, they vary really much, as you can see here. On the left side, you can see the spectra of a cambisol in 3 different conditions. We measure the first spectra in green, the initial water content from the fields. We then look at the maximum water holding capacity of the soil, and when it's completely saturated with water, we take the spectra in blue. And we also oven dry our samples at 105 degrees and record the dry spectra, which is in red. Now, just to make here a quick comparison between our soil types, our cambisol in a fully dry condition has this brown, sometimes yellowish brown color. depending on the amount of goethite in the structure. In comparison, on the right side, you can see the spectra of a Chernozem called Black Earth, intensively studied by Vasily Dokuchaev back in the 19th century. It is described as a soil that has higher granular potential, and it is certainly the most fertile soil type in our region. It has a greater water holding capacity, and you can also see that the absorbance, when you look at the spectra, is higher of the Chernozem compared to Cambisol because of the dark color. Now, this is a simple case when you have two very differing soil types with different color, but there are other cases when our database is composed of hundreds or thousands of spectra representing only one field with only one soil type. Luckily, we have machine learning now to look at them. We also trying to apply commonly used spectral correction methods called EPO, or external parameter orthogonalization, or direct standardization to deal with the water cost changes. Another challenge which we need to mention is the bulk density effect. You can see on the bottom left this graph of the pores by depth. This is our subterranean is measuring this, and you can see when it gets down to the bottom, it touches a really high value in red, more than 250 pounds. When this happens, this also is really visible on the spectra itself. So you can see on the right side, there is this brown spectra, which is exactly at that depth level, 73 centimeter depth. And in this case, if we get this, we usually eliminate it from our data because it's much more diverging than the other spectra. So we don't do modeling with this. It is also important to mention that bulk density is important to look at because for the upscaling of the carbon predictions to megagram or ton per hectare. Yes, one more challenge that we are facing is the topsoil. So monitoring the topsoil comes with very heterogeneous data. So for the use of remote sensing data for topsoil can be really helpful to achieve better results. Now I will talk about a little bit more about upscaling to a landscape. Predicting soil organic carbon stocks on a landscape level. Here is an example from soilgrids.org. This is a system for digitized soil mapping based on a global compilation of soil profile data and environmental layers. There are several hundreds of square kilometers represented here, so it's very large. And you can see Vienna and the Danube, which runs through in black. And on the bottom left side, you can see the Wienerwald. So there's a large forested area near Vienna. And if you look at the legend, you can see that the concentration of carbon in that region is much more higher. Now you can see the same, but here in the legend, you can see that it's about uncertainty. So the trend is when you have a higher organic carbon in the area, you also have a higher uncertainty, even reaching like 50%, which is really high. So for this, luckily we have the work package 3 in PHENET, which is providing us with remote sensing data. So, so with the appliance of this, we can set up surveying patterns based on a stratified random sampling. But currently, we just have results from small scale, farm scale. These are very good results. You can see that for organic carbon concentrations, we have a coefficient of determination, an average of 0.76, which is almost very good. For bulk density, these results are lower, but still significant. Now, soon coming to an end, we cannot forget to mention the use of soil spectral libraries to store and distribute spectral data from our soils. This will help us in the future to provide accurate soil maps and vital information about the containing elements. For the rest of our presentation, I will hand it over to my colleague Pavel to talk about the incorporation of another add-on for our software green device.
Thank you, Akos. So I will be talking about RGB probe incorporation. So RGB probe is different from the spectral probe because even if it's limited to the visible spectrum, it offers a spatial information, which is not possible with the regular spectrometer because it only focuses on one small area. But with the RGB probe, you have an entire image which has a resolution which is higher than just a spectrum. And this offers a potential detection of soil color, even if that's possible to extrapolate the color from the spectrum, but still from the image, it's much easier. And then soil texture, as you can see, extrapolation of bulk density. We can also, as Akos said before, we can also extrapolate it from the insertion force. Also, detection of living organic matter, such as roots or hyphas, or detection of dead organic matter, such as leaves or dead roots. And here you can see that there's an image from not from this probe, but from a mini rhizotron, but this is which we are trying to get. And down you can see the prototype of this probe, which will be tested. Next slide. So, in summary, our initial observations tells us that the probe has a high potential for deep soil surveying, and soil water content or moisture can be predicted with machine learning algorithms and should be used to improve soil organic carbon predictions. Then another observation is that the higher force of insertion potentially indicates greater bulk density, which reflects in the change in the spectra absorbers. Next. Then our current challenges, as Akos said before, we need to test the model with sufficient amount of data, correct for soil moisture, texture, and density, implement stratified random sampling for upscaling to landscape, and combine the RGB probe, which I showed before, with the near-infrared data. Next. Our future opportunities are to be able to detect nutrients and for precision agriculture, identify pollutants such as microplastics or heavy metals, identify abundance of life forms with the RGB probe, and finally the automatization of sampling and soil spectra library contribution. And yeah, thank you for your attention.